So this video is going to be a continuation of our last video on macromolecules, which was missing a very important component called proteins. So stick around. Hi guys, my name is Mikey from Avo Prep Academy, and in this series of videos, we try to cover AP Biology content in a short period of time. But as you guys know, Chapter 5 on macromolecules is a pretty long one. So in our first part of the video, we discussed carbohydrates, lipids, and nucleic acids, but we left behind proteins for this video because I think proteins is the most important part of Chapter 5. So we really wanted to devote a good amount of time to this video so that you have a full understanding of what proteins are all about. So let's begin. So proteins are true polymers made of their monomer constituents. But before we get to the structural part of this, let's go ahead and begin looking at the functions of proteins, because this is going to be really important in setting up what we need to know for this part of the chapter. So in this table, you see that proteins can perform so many different functions, from enzymatic functions to storage, to transport, to defensive, and you have this huge array of functions that AP Biology just throws at your face right here in Chapter 5. Now, of course, many of these ideas you haven't even talked about yet because we haven't talked about the immune system, we haven't talked about hormones nor transport proteins or anything like that. So I want you guys to think about this right now. Remember, in an earlier video, and I'm going to say this all the time, but in biology, form and function are inseparable. And what they're trying to do here is, by giving you this massive array of functions that proteins do, they're implying that proteins can come in many, many different forms. It's this idea that proteins have this incredible structural flexibility where they manifest as three-dimensional molecules that have a shape, that have the capacity to perform specific functions in biology. So we want to keep that in mind as we proceed. So let's start with the structure. Now remember that we always talk about this polymer and monomer thing. So the polymer of proteins are typically called polypeptides, but the distinction between proteins and polypeptides can be a little bit confusing. So we're going to save that for towards the end of this video, but let's just simply begin with monomers, because monomers are relatively simple. The monomers of proteins are called amino acids. And in this diagram, you can see that amino acids are simple molecules with the alpha carbon in the center, an amino group to the left, a carboxyl group to the right. We have a hydrogen on one side of that alpha carbon as well. And you have this really important portion called the R group, or often called a side chain in biology, which then changes from one amino acid to the next. And in fact, in the protein section, we'll see that we have 20 distinct amino acids that can act as a monomer of this long chain of what we call polypeptides. And that idea that the 20 amino acids are the same across all living things really does indicate the probability that we're all descended from a common ancestor. Now let's take a look at these 20 amino acids. Now, this chart displays all of those 20 amino acids in their full glory. Now, you can see that the only R group changes while the remainder of the amino acid remains the same. And as such, the carbon, the amino, and the carboxyl group are often referred to as the backbone, while the R groups are, in fact, the variable side chains. Now, every year that I teach AP Biology, students always ask me, do I need to memorize all of these 20 amino acids and their structures? And the answer is always no, but you do have to know some things about these amino acids and their structures and how that correlates to their behavior. So that's what we're going to start with first. So let's take a look at how these amino acids are categorized, and that will give us an idea as to how their structure can explain why they're categorized the way that they are. So right now, we can largely divide these amino acids into 4 distinct categories, which is nonpolar, polar, acidic, and basic. However, I want to start off by saying something first. Remember that earlier in Chapter 3, where we talked about water and the solubility of substances within it, we learned that nonpolar substances do not dissolve in water, while charged substances and polar substances both dissolved in water. And as such, we can actually take these 4 categories and divide them into 2 main categories, which I think will keep things a little bit simpler to begin. So the first category would be hydrophobic, indicating those nonpolar amino acids, of which there are 9. And then we can say that there are hydrophilic amino acids, the remaining 11, which are either polar or charged in general. And that's because water molecules themselves are polar and they will interact with anything with a partial or a full charge. So right off the bat, we have amino acids that can be divided into hydrophobic and hydrophilic groups, which gives us a really good starting point in describing their behavior. Now, let's take a look at the nonpolar ones first. Remember that hydrocarbons are nonpolar, and right away we can see that majority of these amino acids that are nonpolar only have simple hydrocarbons or benzenes, which are still hydrocarbon rings attached as the side chain. And as such, these amino acids are going to be non-water soluble or hydrophobic. Now, as we move on to the polar groups, you can immediately notice that we have hydroxides, which are polar, as we learned about in functional groups, and we also have some additional amino groups, which, as you might remember from looking at the picture of ammonia and its interaction with water, that these groups can also act as polar substances that can create polarity within the whole molecule. And moving on to the charged amino acids, remember that acids donate protons to solutions, while bases suck up those protons from solutions. And as such, acids, after having given up that proton, are typically denoted as having a negative charge, and bases generally have a positive charge for the opposite reason. Ultimately, what you want to do is to be able to see a chain of amino acids and be able to distinguish which are hydrophobic and which are hydrophilic, because that is going to be really important when you solve some more difficult questions on your unit test as well as the AP Biology exam. Now let's move on to the polymerization process, because once we have the monomers, what we can do is link those monomers. But I want to make a really important point here before moving on. Remember those 20 amino acids that we have? Well, what we can do is to arrange those 20 amino acids in a linear chain, just like we did with nucleic acids. However, one thing to keep in mind is that now we have 20 amino acids to establish a sequence or some combination of those amino acids along a sequence, which then can eventually manifest as a unique structure in the overall polypeptide that forms later. So let's look at the polymerization process. Here we're looking yet again at another dehydration synthesis, which forms a covalent bond. In this scenario where we're looking at polypeptides, those covalent bonds are going to be called peptide bonds. Hence, we get the word polypeptides for having many of those peptide bonds. Now, what happens here is similar to all of the other macromolecules. You have the short polypeptide with 2 to 3 amino acids, and what you want to do is take the monomer and have it oriented in the same direction. In typical textbook fashion, we have the amino group on the left and we have the carboxyl group on the right. And what happens is that the amino group and the carboxyl group will again donate those hydroxides and the hydrogens in forming that water molecule, with the remaining electrons forming a new covalent bond. And you can do this multiple times, and in fact, polypeptides can be hundreds, if not thousands, of amino acids long. So just keep in mind that these 20 amino acids can form a sequence along a polypeptide chain with repetition, because it's not like just because you use valine here that you can't use valine over here. So that is going to give us incredible flexibility in the number of combinations that we can form. In fact, in mathematics, you may have learned about how to calculate combinations. And in the case of polypeptides, if you have 20 amino acids to choose from, and let's say that you have something like 100 amino acids to be sequenced into a combination, well, that is 20 to the power of 100 possibilities that you have, meaning that you have an incredible number of possibilities. Now, not all of those combinations are going to create meaningful polypeptides, but the fact that you have that many possibilities make it much more likely that some of those sequences are going to work. Now, what do I mean by work? One thing that we need to keep in mind is that thus far, we've been talking about a unique sequence of amino acids, which is essentially a one-dimensional set of information. I mean, think about it this way. If you're playing Mario without the jump button and you're simply moving from left to right, then you're really traversing across a single dimension. And just like that, if we have, let's say, the amino end on one side, and say we have methionine, valine, proline, tryptophan, leucine, and the carboxyl end on the other side, going from left to right over here is a simple single-dimensional set of information. However, cells exist in a three-dimensional universe with the X, the Y, and the Z axis all combined into what we call space. And as such, molecules that have functions are also going to have three-dimensional structures. So the first thing that we'll do is give you guys an idea about how this single-dimensional sequence of polypeptides can eventually manifest as a three-dimensional structure. And then we'll talk a little bit more about the four proper structures of proteins in order to understand this entire chapter more fully. So let's first talk a little bit about how one dimension can become three dimensions. In this clip, you'll see that I have a string. Now let's just imagine for a moment that here we have a bunch of nonpolar amino acids. Now remember that in an aqueous solution, like the cytoplasm where these proteins are being created, these nonpolar groups are not going to want to interact with the aqueous environment. Therefore, them being nonpolar are going to start to bend inwards, creating a little shell so that they can face away from water, much like how those hydrophobic tails of phospholipids did. Now, around that shell, we might have some polar groups facing water as to minimize the amount of energy that the molecule needs to spend in stabilizing its position. Now, here's another interesting one. Now, imagine that on one side, we have an acidic amino acid like aspartic acid, and over here, we might have a basic amino acid like histidine. Now, when you take a look at how these positive and negative charged substances interact, they actually attract each other. And as such, this entire polypeptide can bend in order to form another set of three-dimensional structures. Now, there are more chemical interactions involved, but the one thing that I want you to keep in mind is that all of these interactions are happening organically and naturally, not dictated by some hand that's guiding this process, but rather the simple chemical interactions that naturally arise as electromagnetism and polarity and all of these chemical interactions naturally unfold. So now we can take a look at something like lysozyme. Now, lysozyme is a single polypeptide, which is also a protein, meaning that it also has a function. Now, you can see in this image that lysozyme is in fact a single polypeptide chain that's been folded into a three-dimensional structure. Now, in a space-filling model, this actually becomes a little bit more interesting because with the space filled out, we can see that it really does have a three-dimensional structure with domains that can act as active site in allowing this protein to function properly. Now, let's talk about those four levels of protein structures because those are really important in understanding which chemical interactions are involved in each of the structures. These four structures are divided into the primary, secondary, tertiary, and quaternary structures. Let's begin with the primary structure. The primary structure is not really a three-dimensional structure as we might find in nature, but is rather a conceptual one because the primary structure is simply the sequence of amino acids that we have in a polypeptide chain. Now, what that means is if you ask someone, hey, what's the primary structure of the alpha subunit of hemoglobin, then they should be able to write it down on a piece of paper, starting with the N-terminus, with all of the different amino acids within that sequence of 140 some amino acids, ending with the C-terminus. Now, what that means is we don't actually see primary structures existing in nature because it's an idea. Now, you should realize that when those primary structures are thought of in a realistic sense, with those amino acids bonded together, it is going to be that covalent bond or the peptide bond that hold them together. Now, moving on to the secondary structure. This one is a little bit tricky. Now, the secondary structure are common themes and motifs that we see in a lot of different proteins. So this isn't just a structure that exists in a single protein, but these are commonly recurring structures that we see all the time. So here's an analogy to make this a little bit easier. Think about architecture. We have so many different buildings in the world. We have condominiums, we have skyscrapers, we have malls, we have castles, ancient and modern. However, what all of these structures usually have in common are columns and walls, because without columns, the structure would be compromised, and without walls, you wouldn't really have a building after all. So when you take a look at the secondary structures like alpha helices and beta pleated sheets, these sort of act like columns and walls, which are basic building blocks that come in a preset that you can arrange together to ultimately form your three-dimensional structure. Now, one thing to keep in mind here is that the secondary structures are created from hydrogen bonding between the backbones of the amino acids. So there are no R group interactions, but rather the backbones with those amino groups and the carboxyl groups interacting together with those hydrogen bondings. Now, let's talk about the tertiary structure. The tertiary structure is the complete arrangement of amino acids as well as those secondary structures that now form a unique three-dimensional structure. And when I say unique, unique to that specific amino acid sequence. Now, the tertiary structure actually arises from many chemical interactions, some of which we talked about earlier. So I've already mentioned that these amino acids, which are nonpolar, can interact with each other to avoid water, and that acidic and basic amino acids will attract each other. But here we can look at things a little bit more specifically. In this chart, you see all different kinds of chemical interactions that ultimately forms that three-dimensional structure within that polypeptide chain. Here we have hydrophobic interactions, which is in fact those nonpolar groups attracting each other. We have hydrogen bonding, but keeping in mind that the hydrogen bonding here is occurring between R groups, and we also have acids and bases forming ionic bonds. And here we have a very important one. We have what we call cysteine, which is a very unique amino acid with sulfhydryl groups. When two cysteine molecules face each other, they release H2 gas, which is hydrogen gas, while simultaneously forming a covalent bond, which is a very strong chemical bond called the disulfide bridge. And the thing to keep in mind here is that all of these tertiary structures are arising as a result of R group interactions, unlike secondary structures where we exclusively saw hydrogen bonding between the backbones of these amino acids. Now, the tertiary structure is where we have to talk a little bit about the terms protein and polypeptides. Now, the idea behind these terms is that proteins is a functional term given to a macromolecule that has the capacity to perform a specific function. Polypeptide, on the other hand, is more of a structural term indicating simply a polymer of amino acids. Earlier today, we saw lysozyme. I said lysozyme is a single polypeptide, meaning that you have a single chain of amino acids that have folded together into a tertiary structure. Now, that tertiary structure has a function. Lysozymes are part of our innate immune system that protects us from pathogens. So as a result, with that polypeptide having a function, it is also going to be a protein. However, here is where we lead into the difference between polypeptides and proteins. You see, the idea is that sometimes certain proteins, in order to function, require many polypeptide groups coming together. And there we can go on to the quaternary structure simultaneously. You see, the quaternary structure is the combination of many individual polypeptide chains that come together as a protein to perform a certain function. The examples that we see from the book is collagen and hemoglobin. You can see that collagen actually has 3 polypeptide fibers that twist together in order to form a collagen molecule, while the hemoglobin is created from 4 subunits, 2 beta subunits and 2 alpha subunits, which then combine together in order to form a single protein. So in some cases, we can simply define proteins as having a function created from one or more polypeptides, but knowing this difference is pretty important in being able to use the language properly in your FRQ sections later on, so I thought we would flesh that out. So guys, those are the primary, secondary, tertiary, and the quaternary structures. Hopefully that made a lot more sense. The very last part of this chapter deals with a very specific example of sickle cell anemia. And the point of this example is to demonstrate that even an error on the primary structure of the protein can eventually lead to pathology like what we have with sickle cell anemia. So let's take a look at this example very quickly. And on this image, you have what we call a normal hemoglobin. You'll see that on the beta subunit, you have part of the primary structure listed. Now, one thing to notice is that glutamic acid in that 6th position. That glutamic acid, as you guys know, is in fact water soluble because it's charged. However, in a rare genetic condition called sickle cell anemia, that glutamic acid is mutated into valine, a hydrophobic amino acid. Now, as a result, as this polypeptide chain forms into a 3-dimensional structure, this hydrophobic group is exposed to the aqueous environment of the cytoplasm. Now remember that hydrophobic substances do not want to be in aqueous environments, so once these quaternary structures of the hemoglobin molecules form, multiple hemoglobin molecules will start combining together in order to get those hydrophobic interactions to minimize the energy required for their stability. And as a result, we get the crystallization of hemoglobin molecules, which does 2 things. One is that you have the sickle shape forming in the red blood cell as a result of this long chain of crystallized hemoglobin molecules inside the cytoplasm of these red blood cells. And second, because many of the hemoglobin molecules are no longer able to interact with oxygen, you have anemia, which is the inability to deliver oxygen to all the parts of the body that you need. So in this example, we have this amazing ability to take very simple ideas like hydrophobic and hydrophilic interactions, and just simply knowing one or two amino acids, we can eventually talk about the physiology of the disease, not only in the structure of the red blood cells, but also the overall pathology of how it presents within patients. So that about sums up not only proteins, but the entirety of Chapter 5 and Unit 1. Because remember, Unit 1 is going to be primarily based on Chapters 2, 3, 4, and 5. In a subsequent video, I'm going to be doing something very unique. I'm going to be creating FRQ style problems for you guys to think about and watch alongside me, and I will do an explanation video in how to solve those problems. So if you haven't taken Unit 1 test yet, then that's great. But if you already have taken Unit 1 test, well, you can still use this idea for the May exam, so stick around for that video. So if you haven't done so already, subscribe to our channel and click the like button so you can let us know what content you guys want in these future videos. But otherwise, that's it for today. My name is Mikey. Have an amazing day.